Picture a supply room somewhere near Andersen Air Force Base on Guam. Rows of freezer boxes, each one packed with thumb-sized, store-bought mice. A technician threads a small white tablet into each carcass, then clips it to a strip of biodegradable cardboard designed to hook onto a tree branch on the way down from a low-flying helicopter. These bait cartridges are released by the hundreds, drifting into the jungle canopy below. It sounds like the setup to a bad joke. It isn't. It's a real federally funded wildlife control program, and the tablet inside each mouse is acetaminophen, the same over-the-counter painkiller sitting in most American medicine cabinets. For more than 2 decades, the U.S. Department of Agriculture's National Wildlife Research Center has used exactly this method to fight an invasive predator that devastated an island's bird life. Now scientists in Florida are asking a harder question. Could a version of this same trick help against the Everglades' own runaway predator, the Burmese python? The honest answer at this point is maybe in a narrow set of circumstances, and only if researchers can solve a problem that doesn't exist on Guam, a functioning ecosystem that would be sitting directly underneath the bait drop. Burmese pythons are not native to Florida. They arrived through the exotic pet trade, most likely established through a combination of intentional releases and escapes, including, by some accounts, animals that got loose during Hurricane Andrew in 1992, and by the 1980s had a foothold in the Everglades. Genetic and import data assembled by the U.S. Fish and Wildlife Service show that between 1975 and 2018, the live pet trade brought more than 180,000 Burmese pythons into the United States, a pipeline that gave the species ample opportunity to seed a wild population in South Florida's subtropical wetlands. Once established, the snake thrived because the Everglades offered almost everything it needed: warm water year-round, dense cover, and no predator equipped to stop it. Adult Burmese pythons can exceed 17 feet and 200 pounds, reproduce in clutches of up to 100 eggs, and eat prey as large as full-grown deer. Researchers who necropsied one of the largest pythons ever caught in the Everglades, a roughly 17-foot female carrying 83 eggs, removed in 2012, have used stomach content and scat analysis on other individuals to document deer, alligators, and even wading birds among their prey. The ecological toll has been severe and well documented. A 2012 study led by scientists from the University of Florida and the U.S. Geological Survey compared mammal sighting rates along Everglades roadways before and after pythons became established, and found that observations of raccoons had fallen by 99.3%, possums by 98.9%, and bobcats by 87.5% in areas with the longest python presence. Rabbits and foxes had essentially disappeared from python-occupied survey routes altogether. A follow-up study by USGS, FWC, and University of Florida researchers released 95 marsh rabbits into python territory and found that pythons accounted for 77% of the rabbits' deaths within 11 months. Direct experimental confirmation that the snake was suppressing native prey populations, not just correlating with their decline. Against that backdrop, Florida's removal programs have been active, but visibly outmatched. The Florida Fish and Wildlife Conservation Commission's Python Action Team and the South Florida Water Management District's Python Elimination Program pay contractors to hunt pythons on public lands. And the state's annual Florida Python Challenge draws hundreds of competitive hunters for a 10-day event each summer. According to the U.S. Fish and Wildlife Service, roughly 15,000 pythons have been reported to FWC and removed since 2000, with more than 9,000 of those coming from contractors using visual survey methods, real numbers, and real effort, but small next to a population that has never been reliably counted because the snake is so difficult to detect in the first place. The same agency notes that even experienced surveyors in Everglades National Park find, on average, only about 1 python for every 8 hours of searching. Live-baited traps, meanwhile, have largely failed because pythons are ambush predators that don't need to go looking for food in a landscape already full of prey. That combination, a species that is hard to see, doesn't need bait to survive, and reproduces explosively, is why wildlife managers eventually looked past hunting altogether and asked whether a chemical approach, tested somewhere else in the world, might do what humans with headlamps and snake hooks could not. To understand why acetaminophen entered the conversation at all, it helps to look at what happened on a different Pacific island roughly 70 years earlier. The brown tree snake arrived on Guam sometime in the late 1940s or early 1950s, almost certainly as an unintentional stowaway in military cargo. The island's forest birds had evolved with no native snake predators and no instinct to guard against one. Population densities of Boiga irregularis on Guam eventually reached extraordinary levels. Surveys have documented up to roughly 13,000 snakes per square mile in some habitats, and the consequences for native wildlife were catastrophic. According to the USDA, the snake invasion drove the extirpation of all but 2 of Guam's 12 native forest bird species, along with several native mammals and lizards, while also causing frequent power outages by climbing into electrical infrastructure and millions of dollars annually in economic damage. Conventional control, trapping, hand capture, detector dogs, slowed the snake's spread to other islands, but could never touch the population already established across Guam's jungle interior. Starting in the 1990s, USDA Wildlife Services researchers, including toxicologist John Johnston and colleagues at the National Wildlife Research Center, began testing over-the-counter drugs as potential snake-specific toxicants. Acetaminophen stood out immediately. Laboratory trials found that a dose of about 80 milligrams, roughly a child's dose of Tylenol, was lethal to nearly every brown tree snake that consumed it, typically within 48 to 72 hours, while research presented on toxicity to non-target Guam wildlife suggested that birds, rodents, and cats faced comparatively little risk from the doses involved. The Environmental Protection Agency granted the USDA emergency approval to deploy the compound as a pesticide specifically against brown tree snakes. The delivery method solved a separate problem: how to get a dead, drug-laced mouse in front of an arboreal, nocturnal snake without it simply falling to the forest floor where other animals might scavenge it. Researchers and engineers, working with contractors including Applied Design Corporation, developed a biodegradable bait cartridge that snags in the tree canopy on the way down, keeping the bait roughly where the snakes hunt and mostly out of reach of ground-dwelling animals. Field trials measured striking results. An early study published in the Journal of Wildlife Management found that bait take rates on treated plots fell by 83% within 14 days, and mark recapture modeling estimated that tree snake survival on treated plots dropped effectively to 0 over the study period. The program has since scaled to aerial, automated dispensing from helicopters. And in 2015, the Department of Defense recognized the USDA-led effort with its Resource Conservation Project of the Year award for developing a workable, landscape-scale tool against an invasion that had defeated every other method tried. It is worth being precise about what Guam's success actually proves. It proves that acetaminophen is a reliably lethal, selective toxicant for one specific snake species, and that an aerial bait delivery system can put that toxicant where the snake will find it. It does not by itself prove that the same approach is safe or effective in a different ecosystem against a different snake at a different life stage. That's the gap Florida researchers have spent the past 15 years trying to close carefully, and so far only in the laboratory. The first serious test came in a 2010 study published in the CSIRO Journal Wildlife Research by USDA researchers, including Bruce Getz and colleagues, who investigated acetaminophen as an oral toxicant in 2 invasive Florida reptiles, Burmese pythons and Nile monitor lizards. Working with juvenile snakes, the team offered dead neonatal mouse baits containing 0, 20, 40, or 80 milligrams of acetaminophen and found that pythons freely consumed the treated bait and that the compound was an effective toxicant at those doses. A related toxicology analysis by USDA researchers Richard Malden and Peter Savary established lethal dose estimates for Burmese pythons in the range of roughly 263 to 703 milligrams per kilogram of body weight, figures the original CSIRO paper cites directly, and which are considerably higher than the dose that reliably kills a brown tree snake, reflecting real physiological differences between the 2 species. That's an encouraging result. But the same research team was explicit about the limits of what they had shown. The published conclusion states plainly that further investigation into toxicity in adult pythons and monitors is needed, and that any practical use of the compound depends on solving the delivery problem. How to present a toxic bait to pythons while keeping it away from Florida's native wildlife. A challenge the authors themselves describe as enormous. A subsequent literature synthesis on Burmese python biology and management tools, published in the journal Neobiota by University of Florida and USGS scientists, including Kristen Hart, states even more directly that known toxicants, acetaminophen included, are not currently a viable option for python control. In other words, the toxicology works in a laboratory setting on juvenile snakes. The delivery system that made the Guam program viable, a bait dropped where the target species hunts, largely out of reach of everything else, has no proven equivalent in the Everglades. The biological reason acetaminophen might have any future in Florida, it all comes down to python life history. Adult Burmese pythons are ambush predators of large prey, deer, hogs, alligators, and have no reason to investigate a dead mouse hanging in a tree. But juveniles behave very differently. In their first year or two of life, young pythons are smaller, more active foragers that climb into low vegetation and canopy in search of prey roughly their own size, which does include small rodents. USGS and University of Florida researchers who have tracked juvenile pythons using radiotelemetry have documented exactly this kind of exploratory arboreal movement pattern, which is the basis for treating young snakes as the more plausible target for any bait-based approach. The same underlying logic that made the Guam brown tree snake program work in the first place, since that species also actively hunts and climbs at all life stages. The population math behind targeting juveniles is straightforward. A female python doesn't need to survive to die for population control to work. She only needs to be prevented from reaching reproductive age. If enough hatchlings and yearlings can be intercepted before they breed, the removal pressure compounds over successive seasons, even without touching the entrenched adult population directly. It's the same principle behind targeted rabbit and rodent control efforts elsewhere. Suppress recruitment, and the standing population eventually declines even if you never win a fight with the adults. The trouble is that this same juvenile life stage is also when pythons most overlap, behaviorally and spatially, with everything else that lives in the Everglades canopy and understory, which is precisely where the ecological risk calculus gets complicated. Guam's brown tree snake program succeeded partly because of an uncomfortable fact. By the time large-scale baiting began, the island had already lost most of the native forest birds the snake would otherwise have threatened. The ecological damage the program had to avoid causing was, tragically, already limited. The Everglades is nowhere near that state. It remains one of the most biodiverse wetland systems in North America, supporting hundreds of bird species alongside dozens of native reptile, mammal, and fish species, an intact, functioning food web in which almost every animal is a potential scavenger, predator, or prey item for something else. Any strategy that scatters a toxicant across that landscape has to reckon with species that were never part of the original testing. Two groups illustrate the stakes most clearly. The first is the Florida panther, one of the most endangered mammals in North America. Federal and state biologists currently estimate the population at somewhere between 120 and 230 adult and subadult individuals, confined almost entirely to a shrinking range south of the Caloosahatchee River, territory that overlaps heavily with core python habitat. Cats as a group are known to be unusually sensitive to acetaminophen because their livers lack the glucuronidation pathway that lets most mammals safely metabolize the drug. Even modest doses can trigger methemoglobinemia and liver damage in felines. No study has specifically tested acetaminophen bait exposure in Florida panthers, and for an endangered population this small, wildlife managers have been unwilling to run that experiment in the wild. Any full risk assessment for a Florida bait program would have to rule out meaningful panther exposure before deployment could even be considered. And doing that across the panther's entire occupied range is not a solved problem. The second is the eastern indigo snake, the longest native snake in North America, and one of the few Everglades predators known to prey on juvenile pythons. Indigo snakes are themselves federally threatened, and Florida wildlife agencies run active captive breeding and reintroduction programs to rebuild their numbers. An indigo snake that scavenges or hunts near bait mice intended for pythons would face unknown risk from a compound never tested on the species, an outcome that would work directly against one of the few natural checks the ecosystem already has on python numbers. Beyond those two species, researchers have to account for scavengers and secondary exposure more broadly. Raccoons and possums already reduced by decades of python predation. Raptors that might carry contaminated prey back to nestlings with lower drug tolerance than adults. And aquatic food chains. Since aerial bait drops are inherently imprecise, and a share of any large-scale deployment would land in or near water in a landscape defined by canals, sloughs, and marsh. None of this means the strategy is dead on arrival. It means that, unlike Guam, any Florida program would need a far more sophisticated delivery mechanism. One that reliably keeps bait away from panthers, indigo snakes, and other non-target wildlife, while still reaching juvenile pythons in the canopy, before regulators would plausibly approve anything beyond continued small-scale, closely monitored research trials. Florida wouldn't be the first place to learn this lesson the hard way. Australia's decades-long use of sodium fluoroacetate, known as Compound 1080, against invasive foxes, feral cats, and rabbits offers a useful parallel. The poison worked because native Australian species evolved alongside plants that naturally produced the compound and developed relative tolerance to it, while introduced predators had not. A biological head start Florida's Everglades species simply don't have against acetaminophen. Even with that advantage, Compound 1080 programs have documented non-target losses in some native species and, in places, early signs of bait aversion or tolerance developing in target populations over time. A reminder that large-scale chemical interventions rarely stay perfectly confined to their intended target, even under relatively favorable conditions. Florida has also already seen how the public reacts to novel biological control methods, even when the science is solid. When the biotech company Oxitec released genetically modified Aedes aegypti mosquitoes in the Florida Keys, after similar releases had already cut mosquito populations by roughly 80 to 95% in field trials in the Cayman Islands and Brazil, the local response included organized protests, lawsuits, and heated public meetings. This, for an insect, most people are happy to see fewer of. A proposal to aerially distribute a drug-laced rodent bait across one of the country's most iconic protected wetlands. One that would inevitably reach non-target wildlife, including a beloved and critically endangered big cat, would almost certainly generate public and scientific pushback on a much larger scale. Strip away the more dramatic framing this story sometimes gets, and the real state of affairs is this. Acetaminophen is a proven EPA-approved and operationally deployed tool against brown tree snakes on Guam. It is a laboratory-confirmed toxicant for juvenile Burmese pythons in controlled feeding trials. It is not, as of now, an approved or field-deployed control method in Florida, and the researchers closest to the work, including the same USDA and USGS scientists who developed the Guam program, have said directly that the delivery and non-target risk problems remain unsolved. In the meantime, Florida's control effort is a patchwork of imperfect tools working in parallel. Contracted hunters and the Python Elimination Program continue removing thousands of snakes a year. Detection dogs trained to locate pythons by scent have shown real promise in controlled trials, though funding and handler training limit how widely they can be deployed. Radio telemetry. Tracking captured scout pythons back to breeding aggregations during mating season has become one of the more effective tactics in current use, with University of Florida and USGS researchers using previously tagged snakes to lead removal teams to concentrations of untagged individuals. Even robotic decoy rabbits, developed by University of Florida ecologists to lure pythons into detectable positions, are part of the current experimental toolkit. None of these methods, alone or combined, are removing pythons faster than the population can replace them. That is the unglamorous reality behind the Mousedrop headline. Not a program already running at scale over the Everglades, but a genuinely promising piece of chemistry from a different island. A documented toxic effect in juvenile pythons under lab conditions, and a still unsolved engineering and ecological problem standing between that laboratory result and anything resembling Guam's helicopters. Whether that gap ever closes will depend less on the drug. Acetaminophen's lethality to pythons is already established, and more on whether scientists can build a delivery system precise enough to protect a 120-animal panther population and a threatened indigo snake, while still reaching the juvenile pythons hiding in the same canopy. Until then, the Everglades python problem remains what it has been for 2 decades: an invasive,